Now that we've shared how to reduce off-target effects, what are perhaps some of the ways to control for them?
Right. So as one generates data in the research lab and you want to now translate that for manufacturing, there are a lot of different approaches that one can take to begin to understand the off-target changes that occur. So for example, in the products that we're developing, we have to understand the off-target knockouts that occur. And we talked a little bit about the different tools that could be used to find them. And Eric mentioned those, and I won't mention them again. So those are out there, and that helps you understand what they are. And again, I think the control really comes from the system that you use to try to reduce it. So of course we use our DNA RNA guides that we call CRISPR hybrid RNA DNA or chRDNA. So we use that, and that really kind of limits it. And we can control for it, or we can identify it by sequencing. And so that helps us understand. But we also do gene insertion. And so we're inserting a CAR gene, or we're inserting other types of genes, and sometimes more than one. And so there can be like the cross insertion that can happen. You can also, if you have detectable off targets and you know where to look, you can begin to find those things. And so it's really a matter of describing it to try to understand your product. It's a little harder when you're introducing it into the body, that you're introducing the system and you're hoping that it's going to go, let's say, to the liver. It's going to go other places. And so you can look in, mouse models or rat models and begin to understand, where is it going? But in a human, that's trickier because you'd have to biopsy. So I think that it just really depends upon the situation that you're using in order to want to evaluate it. And, to the best of our ability, we use animal models to really understand the potential for these off targets. And it could be, understanding the karyotype and understanding all the different things that can happen. And when we've read over the years, many things that people have found that happen with say CRISPR systems, dependent upon p53 expression and chromosomal rearrangements that are kind of unusual, but that can happen. And so these are just things that one has to evaluate and begin to understand to really capture all that you can for, say, an agency, whether the FDA or ex-US, to explain your product and explain what the product is capable of if it's delivering the system to the body and what it can do. So controlling, I think we kind of covered in terms of how to reduce off targets, but controlling beyond that really has to do with describing the product and what are all of the changes that happen besides the one you intended.
Yeah, sure. That makes sense. Eric, before we move on to taking a deep dive into CRISPR-Cas9, I wanted to make sure that we give you the opportunity to provide your thoughts on how to reduce, how to control for off-target effects.
Yeah, and we know that there's always a certain amount of risk anytime you're doing a gene and editing the genome. But I agree with Steve. I think describing the product as well as possible is what the FDA is looking for. I think it starts with early on preclinical experiments with characterizing the biological pathway that you're targeting and understanding for the gene that you're targeting, what does that once it's expressed to the protein, what's the function, what's the complete function within the cell? So what related to your disease, but are there moonlighting functions that may be that it may be performing other functions? Those should be considered as well to try and limit as much of the unknown as well. I think choosing well-characterized phenotypes with well-characterized pathways with clear phenotypes has been a strategy companies are taking right now to target sickle cell. It's a clear phenotype with the classic sickled blood cell shape. and it's a clear and known effect on the cause for the disease. And so that's a way I think coming into the field and having proof of concept of that, that's where people are reducing risk by picking monogenic targets with well-characterized pathways. I think it's also important once to describe clear safety and efficacy endpoints for your study as well. So as you're thinking about moving into the clinical space, these factors that you've looked at preclinically could be used as biomarkers for clinical studies, and identifying those and describing those and having it in your IND for your clinical strategy is important as well. And for the FDA to see how you're going to monitor those patients over time. As we know, cell and gene therapies have longer follow-up times, with many of them being one-time cures. And so I think the current FDA standard is 15 years for following up on gene therapies. And so need to have a good monitoring strategy to identify any adverse events once you get into the clinic.